let's see. Why are the slides in there? Okay, so just before I start, I just want to mention some disclosures. So I have grant funding from 2 for-profit companies, but nothing that I'm going to discuss today is related to either of these projects. So sleep and circadian rhythms and aging, why should we care about that? One of the reasons is due to the rapid growth in the aging population, not only in industrialized countries, but all across the world. And so these sources from the United Nations show that by the middle of the current century, the number of individuals over age 60 will almost quadruple compared to at the beginning of the century. And surprisingly, that this is not just, again, not just an issue in North America and Europe, but also the population in Asia is aging quite rapidly as well. In fact, Europe is one of the oldest areas with the elder population set to increase by more than 5-fold by the middle of the century to more than 200 million individuals. And along with aging comes changes in sleep. And there are a lot of complaints about sleep that are very common in older individuals. These data are from North American populations, but we find similar things in other aging populations as well. So typically difficulty waking up during the night, waking too early and not able to get back to sleep, waking up feeling unrefreshed. And these are difficulty maintaining sleep in the yellow and early morning awakening in the blue. So these are classic complaints of older individuals about their sleep as they age. We know that there are actual changes in sleep structure as people get older, both the distribution of sleep stages. So here is age, and here are the various stages of sleep. And one very typical thing with aging is that the amount of slow wave sleep, sort of the deepest stages of sleep, declines with age. And then the amount of wake after sleep onset, so episodes of wakefulness throughout the night, sorry about that, increase with aging. If we look at the sleep EEG, we see that one of the reasons the amount of slow wave sleep changes as we age is that the delta activity changes. So this is the delta activity of a teenager with these very high amplitude delta waves. But as people age, the amplitude of delta activity is markedly reduced. And this happens even in very healthy individuals. So in a lot of the studies that we do, we very carefully screen the older individuals so that they are not different in health status from the young participants who we study. So they're not on medications, they don't have chronic medical conditions, and they are otherwise healthy and without sleep complaints. And even in that case, you can see here that the sleep efficiency of even these super healthy older individuals is significantly worse than in young adults. And the number of awakenings throughout the night is significantly greater in the older adults compared to young adults. These are data from just 2 single individuals, a 23-year-old on the top here and a 69-year-old on the bottom here. And you can see, looking at the hypnogram, that this young individual has much more stage 3 and 4 deep slow wave sleep. There's no stage 4 sleep in this older individual and only little bits of stage 3. And that the number of awakenings while brief in this young individual, there's many more, and some of them last for quite a while in this older individual. This is typical of the sleep of even, again, a super healthy older individual. And then if you look at the slow wave activity, again, you can see that there's much while you still have these cycles of slow wave activity associated with non-REM sleep, the amount of slow wave activity is much lower in each of those cycles in older individuals. Now we know that insufficient sleep has a number of daytime consequences. One of those is just sensations of pain and discomfort. And so if you think about some of the typical complaints of older individuals, and think about the fact that they're having this poor and disrupted sleep, then it may be that some of these daytime complaints are associated with insufficient sleep. So for example, if you take a healthy young adult who's sleeping 8 hours a night and just restrict them to 4 hours of sleep a night, they start to complain of joint pain, muscular pain, backaches, headaches, and generalized body pain. All things that, if you think about it, many older adults complain about. Mood is also affected by insufficient sleep. So again, in that same study where healthy young adults were studied both with 8 hours of sleep a night and then sleep restricted to 4 hours of sleep a night, you can see that their optimism and sociability went way down when they were sleep restricted. Their levels of tiredness and fatigue went way up when they were sleep restricted. And there's lots of evidence that attention is compromised, psychomotor speed is slowed, executive function is impaired, and memory is impaired when there's a situation of insufficient sleep. So putting all these things together, what many of the sort of common things that we associate with aging, like pain and in problems with memory and mood, how are those related to the fact that sleep is compromised as we get older? And one of the things that our group is particularly interested is, what role does the circadian timing system play in these age-related changes in sleep? And how can we understand the changes in sleep, knowing that the circadian system is one of the contributors to our ability to sleep? So you're probably all familiar with the two-process model of human sleep regulation that was first proposed by Alex Borbet about 40 years ago now. And that model posits that there's two interacting regulatory systems that regulate cycles of human sleep and wakefulness. There's the circadian timing system, which generates rhythms throughout the 24-hour day in when we're alert and when we're sleepy. And then there's this process called the sleep-wake homeostat, the idea that as you, the longer that you're awake, the more sleepiness you build up. And then when you go to sleep, you dissipate that sleepiness. And if you sleep long enough and well enough, then you'll start out the next day at very low levels of sleepiness and then build those back up again. So we're particularly interested in, are there changes in the circadian regulation of sleep or the circadian system that contribute to these age-related changes in sleep quality? So before I start, sort of delving into those things, I just thought I'd take a few moments to just refresh you on the circadian system and what we know about it. So we think that we have these circadian clocks because all life on our planet evolved in a very strongly periodic environment. So very cycles of light and darkness, cycles of temperature and humidity that are associated with the light-dark cycle. And that any organism that had the ability to predict when those changes, those very regular changes were going to happen, that organism would have an adaptive advantage and then pass that along to their progeny. So the circadian system provides an adaptive advantage that allows the organism to anticipate future events in the environment and to be optimally prepared to deal with those events, whether it's hiding from predators or going out to search for food or conspecific mates, that knowing when those changes were going to happen is an advantage. And the important thing is that this is a property of the internal system. It's not just a passive response to the changes in light and darkness or temperature and humidity. And these clocks are found in organisms at all levels of organization, from unicellular to plants and insects, and even to mammals. And again, they allow the organism to predict future events. I apologize for the barking dog. Things like food availability or light or avoiding predators, and that the animal can then regulate the timing of their behavior in order to take advantage of that. And so there's been a lot of studies kind of showing that this is really the case, many of them in insects, but I particularly like this study that was done in ground squirrels, where it was done by Patricia DeCourcy back in the 1990s. And she actually captured wild ground squirrels, brought them into the lab, did lesions of the circadian pacemaker in some of the animals. Some animals were surgical controls, so they had surgery, but not in their SCN. And then she had some intact controls. And once the animals recovered from the surgery, she released them back into the wild and then went out every week for the rest of the season and tracked where the animals were and what their behavior was. And what she observed is that the SCN lesion animals had a much lower survival rate than either the surgical controls or the intact controls. And when she was out in the field searching for the animals, one of the things that she noticed is that many of them were outside their burrows at night. So typically these animals are day active and they go into their burrows at night. So they're not, they don't become someone's dinner. And that many of these animals were actually either outside their burrows at night or making a lot of commotion in their burrow so that their predators could more easily find them at night. So in mammals, the central pacemaker of the circadian system is located in the suprachiasmatic nucleus. It's shown here in these two red stained areas. This is a section of a human brain showing where the SCN is. So just above the optic chiasm, where information about the light-dark cycle comes in from the eyes, and at the base of the third ventricle. Back as early as the 1980s, it was known that there were prominent cycles of electrical activity, multi-unit electrical activity, if you recorded from the SCN. But later, some really elegant studies done by David Welsh found that individual cells within the SCN could also show these near circadian cycles in electrical activity. So it's not a property of the group of cells, but it's actually a property of the individual cells within the SCN. Okay, why are we not advancing? Here we go. And of course, subsequent research has shown that within the individual cells, there is a molecular timekeeping system that generates these near 24-hour rhythms, a transcriptional, translational set of feedback loops that either promotes or blocks transcription. And since then, we found that there's not only this central clock in the SCN that is then controlling different rhythms within the body, like rhythms of hormone production or rhythms of body temperature, but there are actually functional clocks in individual cells that then form tissues and organs, and not only receive information from the central clock, but receive information locally through temperature or glucose levels or other local signals that help synchronize them. The circadian pattern of expression is widespread throughout the body. These are studies done in mouse, but there's been subsequent studies done in humans showing that many of the genes in our bodies oscillate in at least one of our organs, and that depending on the individual organ, there are anywhere between 650 and about 3,200 genes that have 24-hour expression patterns. There's a core set of genes that are oscillating in pretty much every organ that's been tested. But again, these sort of oscillating genes are widespread throughout the body. Okay, so that was just a quick primer on the circadian system. And now getting into kind of what we know about circadian rhythms in humans. So first of all, there's some key features of circadian systems. The first is that the period or cycle length of the system. So it's typically not exactly 24 hours, but very close to 24 hours. And then is set by some exposure to some signal from the environment. So sort of synchronized to the external environment by some sort of signal from that environment. In most mammals, that signal is the light-dark cycle. And the process of kind of synchronizing to the environment is called entrainment. The interesting thing about light exposure is that it doesn't produce the same effect whenever it happens. It has what's called phase-dependent effects. So depending on the time of day at which you receive light exposure, you can get very different effects on your circadian system from that light exposure. So if you think about a circadian rhythm as a kind of pure sine wave, when we're talking about these things, the period is the time that it takes to get from one point on the cycle to that same point on the next cycle. So typically near 24 hours. When we're talking about the phase of a rhythm, it's just a reference point within that function, which can be the minimum of the rhythm or the peak of the rhythm or the 50% point of the rhythm. It just depends on the particular rhythm that we're talking about, where we use the reference phase point. And then the amplitude, we're talking about the excursion between sometimes the peak and the trough, or other times between the mean and the peak, or the mean and the trough. So first of all, a little bit about the circadian period in humans. So in our lab and in some others, we've used special protocols to actually measure the period of the circadian system in individuals. So this is a paper that we published a while back now, where we had studied the period in about 150 adults. And while the average period was about 24 hours and 9 minutes, you can see that there's a range of about an hour, with some individuals having periods as short as 23 and a half and others as long as 24 and a half hours. And if you look from day to day, so these are a subset of these individuals looking at the time of their melatonin onset each day, and just starting everybody off at the same time point and then following them day by day, you can see that some of the individuals are moving a bit earlier each day. So these are the individuals with shorter than 24-hour periods. Most are moving longer and longer each day, so or later and later each day. So these are the individuals with longer than 24-hour periods. And then there's this one person here kind of in the middle who has a period that looks like it's almost 24 hours. In general, what we see is about 2/3 to 3/4 of people have longer than 24-hour periods, and about 1/4 of individuals have shorter than 24-hour periods. And women are much more likely than men to have shorter than 24-hour periods. This is just showing those same 157 individuals and the amount of adjustment that they have to make each day in order to stay entrained to the 24-hour day. So those individuals whose periods are shorter than 24 hours have to get shifts later each day in order to stay entrained. And those with the shortest periods need to shift by about half an hour each day to stay entrained. And those with the longest periods have to shift earlier each day in order to stay entrained. And again, shifts of about half an hour or so. And then these individuals here whose periods are very close to 24 hours need very little adjustment each day in order to stay entrained. So I mentioned that the light-dark cycle is the signal from the environment that allows, that is the sort of major synchronizer in humans, as in most mammals. The light comes in through a dedicated pathway called the retinohypothalamic tract from the retina and provides information to the SCN that then allows the SCN to kind of know what's happening in the external world. And as I mentioned, our response to light is not equal at all times of day, but it's phase dependent. And this experiment here is just an example of that. So this is an experiment in healthy young adults where both individuals were given the same light exposure, a 1 hour, very bright polychromatic white light exposure. This individual was given the light exposure between about 3:45 and 4:45. This person was given it between about 6 and 7 in the morning. And if you look at the melatonin patterns from before the light exposure, which are the filled circles, you can see that this light exposure shifted the rhythms later, whereas the same duration and intensity of light exposure, but given at a different time, shifted the rhythms earlier. In this case, by about an hour, in this case, by about 1.4 hours. So the very same light exposure can have, again, nearly opposite effects depending on what time within the 24-hour cycle it's administered. And when you sum all those kinds of results together, you get what's called a phase response curve that really is just the time at which the light is presented and the amount of change, whether shifts to an earlier hour or shifts to a later hour. Okay, so that's, again, background about the circadian system. So knowing this, are any of the features of the circadian system changed with age, and can any of those age-related changes help explain some of the changes we see in sleep? So the first thing is there a phase change with age? Well, we know that there's definitely a change in the timing of rhythms. If we look at the timing of core body temperature, it's significantly earlier in older adults than young adults. The same thing is observed with the timing of the melatonin rhythm. So certainly the rhythms are earlier in older adults than in young adults. We also know that their sleep timing is earlier. So then we have to look at the relative timing of their rhythms with age. And one of the things that you can see is that when we unmask the timing of rhythms, in this case, the body temperature rhythm, and align everyone with respect to their usual sleep timing, what we can see is that the young adults here have the minimum of their temperature a couple of hours before their usual wake time. The older adults, they're having, even though the clock time of their rhythms is earlier, the relative timing is closer to wake time. So while the clock timing is earlier, the sort of internal timing with respect to sleep is later. And looking at it in a different way, if we look at the interval between wake time and, in this case, the melatonin peak, what we can see is that the wake time is earlier in the older adults than in the young adults. Sorry. And the timing of the melatonin is earlier in the older adults than in the young adults. But the interval between the melatonin timing and the wake time is actually closer or shorter in the older adults than in the young adults. So in these very healthy older individuals, they have earlier bedtimes and wake times and earlier circadian phases, but they have a different phase angle of entrainment. The next thing that we looked at is whether period changes with age. And again, these are the 157 individuals that we've studied in our lab where we can measure the period. And here is just a scatter plot of those individuals plotted with respect to age. Here's the temperature period. And in a subset, we also had melatonin. So here's the melatonin periods. And you can see that there is no discernible difference in period with age. The period is 24.16 versus 24.12, looking at temperature, or 24.15 versus 24.12 with melatonin. So no significant difference in period with age. So that really can't explain any of the changes in sleep timing or the changes in the phase of entrainment that we see. So the next question is, are there perhaps changes in the response to light of the circadian system as we get older? And I can only have time to present a few studies that have been done on this topic. But there is some evidence that some responses to light may be reduced in older individuals. However, responses to very bright light don't appear to be reduced. So this was a study where very bright polychromatic white light was given. There were no differences in the phase delay responses. There was a slight suggestion that there might have been a reduction in the responses in older subjects in the phase advance region. But again, in the phase delay region, so this is a study done by Phyllis Z's group, again, with very bright polychromatic white light in the phase delay region, and no significant difference in the response of older individuals. When we looked at an intensity or an illuminance response, illuminance responses in young and older adults, one of the things that we observed is, again, there was no evidence that very bright light, that there was a reduction in the response to very bright light, and no difference in the response to very dim light. But in the moderate range of intensities between about 100 and 1,000 lux, there was a suggestion that the older individuals were less responsive to those modest levels of polychromatic light. This is a study done by Deborah Skeen's group, and they looked at short wavelength light, and they found that there was evidence for reduced responsiveness to short wavelength light, at least the melatonin suppressing effects of that light. And there's also been observations that that changes in the lens, so sort of age-related changes in the coloration of the lens in the eye, are associated with sleep disturbances. So again, suggesting that light exposure differences with aging may be contributing to some of the sleep differences. Next, looking at the issue of amplitude changes, there's a lot of evidence of reduced amplitude in older individuals. So these are data from some of our experiments showing reduced temperature amplitude. These are data from Christian Kaioken's group showing reduced amplitude of melatonin and reduced amplitude of subjective sleepiness. And again, these are data from Zurich, where they looked at rhythms in sleepiness and different sorts of performance, and found in the older adults, who are the filled symbols, that they had a lower amplitude in many of these performance and subjective sleepiness rhythms. And when in the few studies that have been done in patients with Alzheimer's disease, it appears that the amplitude is even further reduced in those individuals. So this is a study that was done in young controls, older controls, in individuals with Alzheimer's disease, where there's a reduction in the amplitude of the temperature rhythm in both the Alzheimer's and the older controls. And here, again, you see young adults, reduction in amplitude with healthy older controls, and further reduction in those with Alzheimer's disease. And this is a study of human brain tissue where the individuals were monitored by actigraphy. They were part of a long-term study and monitored by actigraphy periodically as they aged, and then they donated their brains after they passed away. And so there's they found an association between the immunoreactive VIP neurons in the SCN and the amplitude of the activity rhythm in those individuals before they had passed away. So the more cells remaining, the more sort of functional cells remaining in the SCN, the higher the amplitude was, suggesting that there may be changes in the cells of the SCN that contribute to some of these changes in amplitude. Knowing that now there are all these peripheral rhythms, there have been only a couple studies looking at peripheral rhythms and whether the amplitude of those peripheral rhythms changes. This is a study that was done in patients with Alzheimer's disease and age-matched controls. The patients are shown in red and the controls in black. Looking at the rhythms of two clock genes, PER1 and BMAL1, you can see that PER1 appears to be unchanged, both the phase and the amplitude, very similar between the controls and the Alzheimer's patients, although they don't have young controls to compare them to. But looking at the pattern of BMAL1, you can see that the timing of the rhythm in the Alzheimer's patients appears to be shifted and occurring several hours later than in the healthy age-matched controls. So again, evidence for some changes in peripheral rhythms, as well as changes that I showed you previously in some of the central rhythms controlled by the SCN. Okay, so to just summarize what I've shown you, the circadian timing system, one of its roles in sleep-wake regulation is to regulate cycles of sleepiness and alertness, as well as other physiological functions. There is evidence that there are some systematic changes in this system with healthy aging, and even further changes in patients with Alzheimer's disease. And so this may be a target of treatment for both improving physiological functions and perhaps improving sleep-wake rhythms with aging, is by pursuing ways of ensuring, more robust rhythmicity with aging. That may help to boost the amplitude of these rhythms and help improve both physiology as well as waking performance and sleep at night. And so this is I think, an area that we should be pursuing in terms of looking at ways to ensure robust rhythmicity and pursuing further studies of investigating how these rhythms are changing, both how circadian rhythms in the periphery may be changing with age in both healthy aging as well as in the case of Alzheimer's disease. So I will stop there and just acknowledge the Center for Clinical Investigation, where the studies were carried out, which is part of the Harvard Catalyst Clinical and Translational Science Center. Our funding sources, so currently we have funding from the National Institutes of Health and the FAA, but some of the previous work was funded by these other sources. And this is my team with their ugly Christmas sweaters on a few months ago. So again, I'll stop there and take any questions that you might have.